Once again, Sohan, thank you so much for hosting this webinar with us. We are starting the year with a good seminar here, a great start, and the screen is yours.
Okay, awesome. Thank you so much, Flavia, for the intro. So as you might already know, or you may or may not know, I'm Soham Parekh from PerkinElmer, and I represent the MuviCyte system for Western region of US as well as Canada. And you already are aware now that the MuviCyte system has been installed at MBC. So I can't wait to get you guys started, and I'm super excited for you all to use that. So this seminar is basically going to be an introductory seminar in terms of, you're just making sure that everyone understands the capability of this instrument, capability of what sort of assays you can perform, how you can tweak your analysis, imaging, so that everything is compatible with the MuviCyte, as well as how can you best utilize the MuviCyte system for your own workflows. So again, I'm super excited and thank you so much for being here. So this is, just making sure that everyone can hear me and see my screen. And
Yes,
okay, awesome, great.
perfect.
So awesome, great, thank you. So as you can see, this is how the MuviCyte instrument looks. And just to walk you through all the features and capabilities of this instrument, I just wanted to start off with some basic understanding, because we have companies at MBC. Some companies are already established in their cellular imaging workflows. I've talked to several of you guys, and some of the companies are right now in the progression of starting their cellular imaging workflows. So I just wanted to make sure that everybody's on the same page. So I'm just going to start very introductory introduction on what is live cell imaging and why could this instrument be important for your workflows. So, this, when I was a researcher, I always used to take all the immunofluorescence images or just images at static time points, such as, you see here. So I would take images at 0 hours, 24 hours, 72 hours, 36 hours, right? And I felt pretty happy with my workflow back then. But what I was forgetting, or what I could have actually done, is looked at the blind spot in between those different time points. So for example, if you are adding some drug, incorporating some drug, and you are seeing the effects of the drug effect at static time points such as 24 hours, 48 hours, you're actually missing out on a lot of details in between what happens between the 0 time point as well as the 24-hour time point. And the second thing, what I also was doing, because I was taking, for example, let's pretend that I was working with three different time points, 24 hours, 48 hours, and 72 hours. I had to prepare my samples thrice. So, not only I was doing everything in triplicate, but I had to prepare those triplicates three times so that I can accommodate all the time points. But with this live cell imaging technology, you can actually circumvent that, and you only need to prepare one triplicate. You can monitor it throughout, and you can all get all the physiological details from all the time points. So it's a very cohesive story that you can acquire from live cell imaging, and that's why it's really important that everyone understands the capability of not just this instrument, but also how powerful the live cell imaging field is. So with the live cell imaging, you can not only understand when something happens, because you would be getting all the information about different time points. You can also understand how fast it happens. For example, if someone is working on wound healing, you can even get the rate of the wound healing, rate of the migration, how the cell confluency is going up and at what time, particular time point. So you can get a lot of more details. And even if, you have some susceptible cell types, you can even understand by what mechanism your progression of physiology of cells is going on. So a lot of details to be, assessed with the live cell imaging, and that's why it could be really, really important that, I would really encourage you guys, if you're not already performing any live cell assays, just start performing that, and it's really, really going to open up a lot of possibilities for your group. So this slide is actually, this particular instrument is installed at MBC. This is actually same MuviCyte that has been installed. This is your own incubator. And Greg from your facilities team was kind enough to send me a picture of that. So as you can see, all nice and pretty, it's already installed, and you can see a plate which is already loaded on this instrument. And what I just wanted to highlight from this picture is that you can perform live cell imaging in different variety of ways. But MovieSite actually fits inside the incubator. So that's, a very, very big advantage, because then your cells would be exposed to direct environment of the physiological conditions that the cells are supposed to be in. So let's just see this plate. You see this is an open interface, so your plate is directly exposed to the outside environment of the incubator. So your cells are literally sitting inside the incubator at the same conditions that they are supposed to be, and also getting imaged at the same time. So you can perform all the imaging experiments worry-free without worrying about, how your cells will interact or if the behavior of cells would be changing or not. You don't need to worry about that, because not only this instrument goes inside the incubator, it is also an open stage interface. And the second thing I wanted to highlight is, because we have this open stage interface, your samples, because they are directly exposed to the environment, if you have any susceptible cell lines, such as, someone is working with primary neurons or some primary other sort of stem cells or iPSCs that are really susceptible or sensitive, you can make sure that they are always at their physiological conditions, and you don't need to worry about, your cells misbehaving because of the environment they're exposed in. So that's a tremendous advantage that you all will be getting with the MoviCyte. In terms of its operating environment, it's pretty standard incubator conditions. So it goes from 5 to 40 degrees Celsius, from 20 to 95% humidity. And one great feature about this instrument is also Perkin-Elmer was, smart enough to install UV lamps in the inside of this microscope. So what I mean by that, so just see that, you already can see that your microscope is sitting inside the incubator, and we all know that the incubator conditions would be very warm as well as very humid. And sometimes you have to take the instrument in and out because you need to use some sterilization cycles on the incubator, and that would gradually it would start contaminating your inner components of your instrument. And to prevent that, or to limit that, we have actually installed UV lamps towards the inside, so it is always protected by that. And this is again my one of my favorite features about this instrument, that you can pretty much use it for any sort of use. So this is kind of an experiment people were running with a microfluidic chip on this sort of slides, and you can literally see that they are taped on top of the microscope stage, and we are still able to perform long-term live cell imaging experiments from this sort of setup. So not only it is susceptible because of its open stage interface, the images that you get from this microscope are really stable. And because of its very, very small footprint, this is actually one of the smallest incubator-based microscopy systems that is, existing currently. And because of its smaller footprint, you would have a lot of room to accommodate all sorts of different vessels or assemblies or other components of your experiment if need be, like you can see from this particular experiment, and you can still perform your imaging assays as conveniently as you want. So this is again, movie site, how it looks. You can also see a plate on top of that. And one more thing I wanted to point out, that, since you would be starting to use this instrument, you would also need to, just have an idea about how you can operate that and everything. So everything will be covered in the training, which will take place in the next week. And I would have some follow-up details on that as well. But for now, I just wanted to highlight that, this instrument would be connected to this control box, which is sitting outside the incubator. So this is literally underneath the incubator that you guys have currently. And this pretty much works as an on and off switch. So for some reason, you had to take the instrument out or something, you would use this control box to turn it off. And then this pretty much works as an on and off switch. One great feature about this instrument is also this instrument can be a benchtop system for a microscope. So this literally can also be a tabletop microscope. However, because you have a lot of users and a lot of companies who are actually really excited to get started on this, I think this is pretty much going to be used for all the live cell workflows. So you would not be using it on a bench. But if that was the case, and if you ever needed to use that, we also have a lid which goes on top of this microscope, and it pretty much works as a conventional microscope. Does anyone have any questions so far? Any questions so far? I cannot see the chat. So guys, if you see any questions, please let me know, or feel free to interrupt. It is Yeah, feel free to interrupt at any time if you have any questions. Thank you. So if there are no questions right now, I'll keep going. Um, with the movie side, not only you can get these flexible imaging modes, but you can also get really nice pictures. And it is equipped with three different filter sets, so you can have RGB channels, meaning red, green, and as well as blue fluorescence channels, as well as bright field, and you can create these beautiful images from the movie side. And this is something I wanted to highlight because you are really going to be very happy with the images that you get, and not only for the IF, but even for the all the live cell experiments that you would be performing with this instrument. And this is just an example of we have created this overlay images, and again you can just double check that how great the images look from the movie side. And just to add on to that, why we can acquire those great images is because we have equipped it with a very nice camera, as well as three different objectives, which are all long working distance objectives, and they have a high NA, numerical, high NA. So you would be getting very good and sharp images from this particular instrument. As you can see, you have green as well as red as well as blue channels that you can facilitate with this instrument, and you can perform imaging from 4x, 10x, and 20x long working distance objectives. And the long working distance objective could also be pretty useful if you have any thick plates. For example, if you are not using optical quality plates, you don't even need to worry about imaging that particular plate on a movie site, because of this long working distance objectives, the autofocus is going to still set pretty well. And from my own experience, I've observed that the autofocus is really nice of this instrument, so you would not be disappointed with that. And that is partly because of the long working distance objectives that we have equipped it with. And coming back to the flexibility mode, so one of my favorite features again, excuse me, that it is super flexible in terms of what you can image and how you can image that. So on the top left, you see, we have selected different fields of views within a single well. Just to give you an idea, you can select hundreds of fields of view within a single well, and then it is very flexible. So if you had to select like 3 or 5 different positions in one well, 100 in a second one in the third one, you can still do that. So it's a lot of flexibility that you get with this instrument. And particularly what that could be really useful for is, if someone is going to use this instrument for confluency analysis or cell proliferation analysis, what you can, conventionally what I used to do was I used to get a stitch right in the center of the wells, and then I would get the confluency readouts from that. And I would, try to create a montage or a stitched image of 2 by 2 in the center, and then I would interpret that as the confluency readings. But with the MuviCyte, what you can also do is not only that, but you can actually select one single field of view in different locations. So you can just select all the field of views with a lot of freedom wherever you want. And that would actually give you a more sophisticated readout of what is actually going on. Because, when you seed your cells, sometimes they stay in the center, sometimes some of the cells are more dispersed towards the edges of your wells. So to eliminate that, to eliminate those sort of outliers, you can actually select them pretty much everywhere in the well locations, and just get the confluence readouts from that. So that would be more physiological relevant. In terms of its autofocus, we have a different autofocus modes, and it's really useful to avoid phototoxicity and photo bleaching, as well as to increase the speed of measurement. So I would be covering everything in the training sessions. Again, I'll be talking about the training schedule at the end of this presentation, but during the training, we'll also go through how you can set up different autofocus modes so that you can actually avoid a lot of phototoxicity as well as photo bleaching. So people who are new to the live cell imaging world, phototoxicity is basically if you emit the light to the fluorophore, your cells, because of the fluorophores present, your cells would start producing reactive oxygen species, your cells would start exhibiting some of the phototoxicity. So with the MuviCyte modes, you can actually eliminate that a lot. Photo bleaching is, your dye basically bleaching out. So if you had example, if you had a dye, if you emit light several times on the same dye, what would happen is it would basically reduce its capability to emit the light from the same fluorophore. So you can actually avoid all those things by using different focusing modes as well as autofocus modes on the MuviCyte, and you can just make sure that your cells are all nice and happy. So even if you wanted to, perform the imaging experiments for a week, 4 days, 3 days, with the fluorophores, you can easily do that with the MuviCyte with the help of all these different autofocusing modes. And again, we'll be going through all that. So you would be all ensured that you can use that properly. And in terms of offsets, you can even create custom offsets. So we have a feature where you would, the software would pretty much use its autofocus on a bright field channel, and then you can actually customize the offset for the fluorescence channel. So let's say that your fluorophores are like super sensitive and you would like to not emit the light during autofocus on the fluorophores. You can just emit the light using the bright field channel, and then you can customize the offset so it's not going to use those autofocusing modes in the fluorescence channels when it's going to image. So again, all of these things would be explained further in the training sessions, but just know for now that you would have a lot of flexibility in terms of how you can set up different focusing modes so that you can avoid any sort of photo bleaching and phototoxicity. Any questions so far? Okay, seems like we don't have any questions so far, so I'll keep going. With the MuviCyte, not only you can get all these flexible imaging modes, but you can also perform Z-stacking with that. So if you had any spheroids or organoids or some, tissue that you would like to get some Z-stacks off, you can get that. With the limitation of wide field microscopy, you can still create some great Z-stacked images with this MuviCyte, and our stack depth can go up to 350 microns. So that's pretty substantial actually when you think about that. So this particular example is, of a spheroid, and I'm just showcasing how you can utilize the Z stacks of this microscope to get your spheroid images, and then you can pretty much create maximum projections out of those images and get all these details from the spheroids as you wanted. And of course, you can create all the Z stacks from all the fluorescence channels as well as the bright field channel. And with the image stitching, again, just an example of how great the images look from the MuviCyte, but also, this is also an example of how you can perform the stitching with the MuviCyte. So we do have different vessel holders, so you would be able to accommodate your slides on top of the MuviCyte. And if you wanted to just do some slide scanning, you can still do that with the MuviCyte. And we have a lot of different stitching modes, and the stitching can go up to 99 by 99 images. So that's almost 1,000 images compiled together. So it's a huge stitch that you can create with the MuviCyte. And you can also do whole well stitching. So perhaps you had a 6 well plate, or 94 well, 96 well plate, or 48 well plate, and you wanted to do the whole well stitching. You can still do that. There's just a button for that. So you just click on that. It will auto set its own stacks, or or the stitches, and then it would pretty much create all the well stitch images for however wells, how like number of wells that you have selected for. And you can also perform the stitching not only with all sorts of well plates, but also with all the objectives that we provide you. That is 4, 10, and 20x objectives. So with this information, I just wanted to highlight on some of the analysis features that you would be getting. So each MuviCyte user can ask for it, and I'll be actually happy to provide you with this software. So basically you can load this software onto your computer and do some preliminary analysis if you wanted. So this MuviCyte analysis software suite is good for growth curves. So if you wanted to monitor any cell confluency or something, cell proliferation or something of that sort, you can use that mode to analyze those sort of details from your cells. You can also get scratch basic assay analysis done from that. So if you had any scratch tests going, you would have ability to just pretty much quantify all the details from your own computer if you wanted. You can do fluorescent cell counting from this software suite. So if you had any just need of cell counting, which this is something that I pretty much was working on during my research. I just needed to count the number of fluorescent cells, so you can easily do that. And this can also be used for any sort of live dead analysis that you are doing, or if you are working with any transductions or transfections, you can still use this mode in order to get all these details, all the quantifiable details from your workflows. And you would also be getting a copy of this movie maker. So what this movie maker does is basically you can create single movies, or you can create these sort of matrix movies. And these movies can go up to 9 frames by 9 frames. So this is an example of 2 frames by 2 frames. And you can pretty much just turnkey software, just make a couple of clicks, and you would have these sort of movies right away from the software itself. And it's pretty helpful when you have to, compare different conditions. For example, you are doing a 7-day workflow of, live cell imaging. You're adding some sort of drug, and you want to see the effects of that particular drug on your cells. Not only you can quantify those details, but you can also present them in this way, comparing all the videos together, and that would also give you an idea about what is going on with your cells. So that's really, really useful. So the accessories that have been provided to MBC are all of the above. So you have access to a scratcher tool. So, if someone is performing any scratch test assays or would be interested in performing scratch test assays, we have the scratcher, which would be producing 96 even scratches in your 96 well plates. And then we also have a scratch software, which can quantify all the physiological details from a scratch wound assay. We also have a spheroid software. So if someone is working with spheroids, we also have a dedicated software for that, which can pretty much quantify all the spheroid size, morphology, the area, etc. Also the circularity of the spheroids. And if you just wanted to monitor any sort of 35 millimeter petri dishes, 60 petri dishes, or any sort of slides or tea flasks, you also have accessories for that. So you can just pretty much load those onto the MuviCyte and monitor the cell cultures, across the periods of time that you're interested in. Any questions so far? Okay, perfect. No questions so far. So keep going on. Stem cell monitoring. So, you can perform all the monitoring studies from MuviCyte, as you would understand from a live cell imaging experiment. But the differentiator being the MuviCyte images are going to be super stable. So you would not see any sort of jitter in these images. And I think these are, one of the best sort of stable images that I have ever seen in an incubator microscope, or even like an on stage microscope. The stability of the stage is really, really super impressive, and you can pretty much make sure that all the cells are in focus all the periods of time. This is an example of stem cell monitoring that someone had done. These cells were pretty much grown for 3 days. The plate was actually taken out from the microscope twice for culture media exchanges, and you can still see, you won't even notice if there is any difference in terms of what you're seeing on the frame. And I have actually done the same sort of experiment with some of my researchers that I'm working with for a demo. And they were also really, really impressed by how stable the images look from the MuviCyte. So this is also what you would be getting, and this is also something that you should consider while you are using the MuviCyte. So all the images and all the videos, time lapses are going to be exceptional and publication quality right away. So you don't need to do any image processing on that, and you just don't need to worry about your cells moving in and out, because if you imagine, if you're working with a spheroid and if it's going in and out of the frame, then if you quantify those results will not make perfect sense. But with the MuviCyte, you would not need to worry about that at all. This is an example of suspension cell culture, and that actually I had performed in my own lab. So I was working with B cells, and as you can see, the B cells are sort of aggregating when I added some conditions to the cells. So basically, this was done for 4 days. But the thing that I wanted to point out is the autofocus is on during the course of this study, as well as this is a 4 by 4 stitch image, and I was working with a 96 well plate, and I had at least 48 wells loaded with cells. So this was not just the one well that the instrument was imaging. I was monitoring all across the plate, and you can still see that the images are like super sharp and stable. And that's because we have optimized the speed of this stage moving. So basically the stage is optimized so that it moves slowly. So even if you have any sensitive sort of cells such as suspension cells or spheroids, it is going to stay right at place, and it's not going to fluctuate or deviate its position, and you would not see any differences when you actually get the raw data out of the images. And now the interesting part, right? So key applications. So I think, from the previous slides that I had pretty much talked about, you would have some sort of idea in terms of how you can utilize this instrument for your own workflows. But I just wanted to give you a couple of examples on how people have used this in the past. So this would also open up some possibilities that, you may or may not have thought about that you can use this instrument for. So you can use this MuviCyte for cell health as well as viability. You can perform any transfection, transduction assays. You can perform scratch wound assays, and I would have a dedicated slide to explain what I mean by that. You would also have a software for spheroid analysis. If you wanted to monitor spheroids over the periods of time, you can still do that. You would have accessibility to perform neurite outgrowth if you are examining chemotaxis or just overall cell migration or cell movement. You can perform all sorts of studies. So once you start using this instrument and you have a strong hold on that, you would just think of a lot of different ways in terms of how you can utilize this for your workflows, and this would open up a lot of different project possibilities also. So really, I cannot wait for you all to get started. So super excited for that. So this is an example of cell proliferation. So this is just pretty much basic assay that you would be using this instrument sometimes for to just monitor the cell proliferation. And we have a dedicated software for that. So it's a very 1 or 2 clicks, and you would create all these simple masks. And these marks are really effective, super easy to perform. And then you would be able to quantify all the details about your cell proliferation. You can also perform cell health as well as viability. So this is an example where increased concentration of drug was added towards the right side of cells, and all the green cells that you see are exhibiting cytotoxicity. So you can get the cell confluency readouts as well as the cell death readouts from the MuviCyte dedicated software, and pretty much perform all the live dead assays as well as perform the cell health and viability assays from that. You can also perform transfection or transduction efficiencies. So if you are, optimizing your workflows and you wanted to check in like what is the exact time point where I have to perform the transfection for, then this is really useful. You can perform all the studies on the MuviCyte, and because of its great autofocus modes, you would be totally be very confident with using this instrument for all these studies. Pretty much in transfection study, what you would need to accommodate for is like your cells would not be having any fluorescence in the beginning, and then it would express the fluorescence at the end. But I think with the autofocusing modes, you would be easily be able to do that with the MuviCyte, and I'll be happy to assist you set up your protocols if you need it. So yeah, again, transfection, transduction efficiency studies if you wanted to, and the scratch wound assay. So this is really great. I used to perform the scratches on my own using like a pipette tip on a 24 well plate, and I'm sure like some of you might have already tried that or done that in the past. And you must agree with me that it's not reproducible. Like I pretty much dedicated almost 8 months working on different simultaneous projects, one project being just a scratch test assay, and I could never figure it out perfectly. My scratches would all over be the place, or be all over the place, or there's like just increased amount of weight that you put on the pipette tip, and that pretty much changes your physiology of the cell migration. So to circumvent everything like that, you have a dedicated scratcher tool now. So if you are performing any scratch tool or scratch wound assays or thinking about performing one, this is an amazing tool. So What you can do with this is you can seed your cells in a 96 well plate. Once they are confluent, you just put them on this tool, make the scratch. You have 96 dedicated reproducible scratches throughout your plate, and then you throw it in on the MuviCyte, and there is a scratch wound um, um, dedicated scratch wound analysis software that we have, and you can quantify all the details. So you can quantify not only the cell confluency, you can also, understand what is the difference, what is the distance between the wound site, how fast are the cells migrating, and so on. So this is really, really amazing and super simple thing to use. So, I really, really would be happy to assist you with that. And I've actually spoken to several of you. I think two labs were at 733 location. So, if anyone is needing any assistance with the scratch wound assays, just let me know and I'll help you set it up. The second thing you can also do is, if you had any spheroids or organoids, particularly for spheroids, actually, this we have a dedicated software for that. So not only you can monitor your spheroids across the time frames, but you can also understand different physiology, such as its diameter, its perimeter, area, volume, as well as circularity of the spheroid. So we also have a dedicated spheroid software for that you already have. So just feel free to use that, or ask me any questions if you had any sort of spheroids that you're working with or needed any assistance with that. So to summarize, MuviCyte is a pretty impressive tool that is going to be sitting inside your incubator all the times, and you'll be performing live cell assays with that. And you have three fluorescence channels, red, green, and blue channels, as well as bright field channel in the MuviCyte. You have 4x, 10x, as well as 20x long working distance objectives. You have so many different autofocusing modes that would assist you avoid any photo bleaching, phototoxicity. You also have a dedicated image quantification software for preliminary analysis. You have a movie maker software, so you would be able to compare all the live cell assays across the time. All the data that you get from MuviCyte files are universal format. So basically, they're open data format, and that can be implemented on any sort of software. So if you are dedicated, if you are already using some sort of imaging software, such as MRS or ImageJ, then you can continue doing that if you wanted. You can just export all the images as is, and they are all open data formats, so they can easily be accommodatable everywhere else. You can also perform Z-stacks and flexible imaging modes with the stitching capabilities of this MuviCyte. So if you had any 3D cell cultures, if you had spheroids, organoids, you can easily do that. Even if someone had any microfluidic platforms, I'll be really, really happy to help you set it up. Because of all these flexible imaging modes, we can also perform all sorts of microfluidic imaging on this MuviCyte, and you can perform all the live cell imaging assays on, like, let's say, organ on a chip platforms. And one last thing to remember from this particular introductory webinar is you are really going to be amazed with the image quality as well as the stability of the stage. So whenever you are setting up some protocols, and if you are considering which instrument to use, just you have the information that you can use all this information and then decide like which would be more suitable tool for you guys to use for the live cell imaging experiments. So with that, I just wanted to, I would have some room for the questions and answers towards the end, but I wanted to highlight that I would be conducting virtual trainings next week on the 19th, as well as 20th, as well as 21st. So those sessions are going to be the same sessions on all 3 days. So feel free to sign up for one session. And then if you are, open, your schedule is open for more than one time slots, please leave me a note with your preferences. So I have actually set up a dedicated registration link. So I'm going to be posting that into the chat. Let me try to do that. So you can use this. Okay, it's not letting me do that. Let me try once again. One second, it's loading. Okay, so this is the sign up link for the training. So depending on what time you are available at, please leave please mark your presence if you are going to attend any of the training sessions. So what I have allocated the fields for is pretty much you would input your name as well as your name of the company as well as email, so I can reach out to you to set up an appointment for the training. And it's going to be a training across different MBC teams, so there'll be multiple people attending the training. And due to the COVID restrictions, I would try to split you guys into even number of groups. So I would try to split into like even number of people for each of the training sessions. So not only you can benefit from the personalized training, but also we can be wary of the COVID restrictions. So if you can click on that link and start signing up for the training sessions, that'll be super helpful. So that way we can, easily set up different sessions, and then we can go from there. And if you had any questions, you can reach out to me on my email, or if you wanted to, just email me with your preferences of the training sessions. I'm also posting my email in the chat, so you can just email me that information, and we can set up an appointment for you. Um, so just one thing that I wanted to show you was, how you register for the training session. So let me start sharing my screen one more time. 1 second. So this is how your screen is going to look like when you start signing up. So once you open the Excel sheet, it would be on the viewing mode. So you can easily click on this editing mode. And once you click on that, you would be able to edit, and you can add your name and set up your preferences. And first preference is, which is your preferred time slot. So for example, if I wanted to set up, Friday as my first preference, you would add Friday here in the first preference. Then I would ask you to also put your second and third preference if you're available. So, depending on how flexible your schedule is, I would encourage you to at least put in two different preferences so we can, just keep the numbers even across the board. So I see people are signing up, so that's great. Thank you guys for doing that. And while you do that, just wanted to double check if anyone had any sort of questions for me or our team. Okay, so it seems like everybody understands MuviCyte pretty well. Everybody's super excited to join the training session, so it's amazing. So I see people are still signing up. That's great. And I'm just going to be around for 5 more minutes. If you had any sort of questions, feel free to type it in the chat or just unmute yourselves and ask the questions right away. I'm here for you guys. Yeah, thank you, and thank you so much for attending this webinar. And I hope that this was really helpful for you. And hopefully the MuviCyte would be a great tool, a great addition to your workflows. And I'm really excited to get you all started.
Sohan, thank you so much for your very informative webinar. We are super happy to have PerkinElmer supporting us and the MuviCyte now on site, and I believe everybody is super excited to start using it. I will also send the email with the link for the training, so people that couldn't attend the webinar today, they will be able to register as well.
Awesome, great, thank you.
And thanks so much everyone for attending. First event of 2022. So we are starting with a very good seminar here. Thank you.
Thanks for having me, Soham. Take care.
Thank you, Grant.
Thanks, that was great.
Thank you. Right, I think we can stop recording now. And I think it went well. I think people are super excited. I know like several people reached out to me directly saying, "Oh, when can I set up the training?" So I'm sure a lot of people would be, really, really eager to get started. So, I'll conduct the training sessions next week, and then pretty much they would have a strong hold on, how to get started. And hopefully even through the virtual training they would be super confident to start using that. So yeah, just yeah, excited to get going. Yeah. All right, awesome.